I teach AP Biology. Let's play Are You Cooked? My first question is really 3 questions. Question number 1, what is this whole molecule called? Question number 2, what is this bond labeled number 1 called? And question number 3, what process formed that bond? So again, 1, what is it? 2, what is that? 3, how did it get there? Number 1, it's a protein. How do I know? That nitrogen right there. If it was a nucleic acid, it would have nitrogen and probably phosphorus and maybe sulfur as well. If it was a carb or lipid, there would be no nitrogen there. Next question, what is this bond called? The answer is a peptide bond. We have 2 amino acid residues, which are the building blocks of proteins, the monomers of proteins, okay? And in between 2 amino acids, you will have a peptide bond. Last question, number 3, how did it get there? There's a process called dehydration synthesis. Dehydrate, we had to remove a water, and by water we mean like an H, another H, and an O, right? We removed water to synthesize the larger molecule, or in this case, the dipeptide. That was a lot of chemistry, so let's end on an easier note. What organelle makes that? Ribosome. Okay, my biology 1 students do have to know that much. Follow up, where can the ribosomes be located? We can have them free floating in the cytoplasm, or we could have them located on top of the rough ER. If a protein is destined for export, where is it likely going to be made? Probably the rough ER. That goes back to that whole endomembrane system thing. Here's a nice little visual of what that looks like. It's not perfect, but it gets the job done. We've got our little proteins being formed on the ribosomes here. Then we're transporting them through vesicles to the Golgi, which is going to package and modify them and then export them out of the cell. So this was an easy one. Know your proteins, guys. Know them.